Hello, everyone, and thank you for coming to this presentation. My name is Tania Pereira Chilima, and I'm the Chief Technology Officer at Univercells Technologies. Today, I'll be talking to you a little bit about how we see technology innovation enabling developers or gene therapy industry to bridge the gaps in capacity. So today I'll introduce I'll introduce Univercells Technologies. Then I'll talk a little bit about the capacity needs in the gene therapy field, and then talk about how we see technology innovation bridging those gaps from both technology design through what we call chaining and intensification, as well as technology performance. Then I'll close with some concluding remarks. So Univercells is a subsidiary of Univercells Technologies, funded last year with the objective of revolutionizing virus manufacture through technology innovation by using the principles of intensification and training. And our leading products are the Nivaline platform and the ScaleX bioreactor. And I'll talk about them a little bit more in this presentation. So, as we know, there are a number of clinical trials for gene therapies currently ongoing. Here is just a snapshot of a few clinical trials that are ongoing using AAV. What we see here is that both the dose size used and the prevalence of the different diseases varies significantly, which means that we end up with a huge difference, so several logs difference in the annual demand required for different indications. So when we map a few of these indications across the different technologies that are currently available, what we see is that for technologies with lower annual demands, so more like ophthalmology kind of indications, we see that flasks or manual-based technologies could still be the optimal system to use. But as we move up, we see that for very large annual demands, the stir tank bioreactors seem to be, or using suspension cell culture, seem to be the optimal technology or the most scalable technology. Even so, of course, there are some assumptions made in this slide, but even so, for certain indications, we see that multiple stir tank bioreactors will be needed in parallel in order to support the annual demand. And this is just kind of a mapping of the current technologies available in the market versus the annual demand for different gene therapy applications. But when we take stir tank bioreactors into account as a technology for large scale manufacture of gene therapy products, one thing that is important to consider is the impact on the facility itself, because these systems do produce a large volume that needs to be processed through midstream and then downstream. So then we just looked at, how is the volume generated? So the volume containing the cells of the product generated and how it moves across different stages of the process. And what you will see is that once you get to the cell culture and virus production stage and leading to the midstream process, which is when you're clarifying the product, this is really when you're having the largest volume of product being moved around. And this, of course, has facility implications. And we see that as the key hotspot for intensification and chaining, basically. So the linking of different unit operations together and automating them to really reduce the volumes being transferred. And this is important because, as I mentioned in the previous slide, we are looking at potentially needing multiple trains, so production trains in parallel, which means that if these volumes are not somehow reduced, the impact on the facility footprint would be really large, and leading, of course, to capital expense, so impact on the capex as well. So when we looked at technology innovation, we looked at two key axes. So the first one is to increase performance, to bridge that gap in capacity. But the second one is also to reduce the volumes being processed, so to reduce the volumes being transferred. And that's achieved through intensification and chaining. And that's why you see here this arrow going towards the left-hand side, which is we want to look at a technology that both increases performance while reducing the overall footprint of the operations. And this is what I'll discuss on the later versions of this presentation. So starting with the first axis, which is basically reduce the footprint of the operations, I'll talk about how we use chaining and intensification to achieve this goal. So, as I've mentioned before, a traditional manufacture of gene therapy products or biologics relies on standalone unit operations and batch processing, and having large holding tanks in between them and having movement of volumes across unit operations. What we are looking here is to look at two principles, intensification and chaining, that I briefly talked about, to both reduce footprints and also to not only reduce footprint of unit operations, but also reduce the size of the surge tanks. They're linked them together in order to have an impact on both footprints and capex, footprint, capex, and opex. So, for us, process intensification starts in the bioreactor through biomass immobilization. So what we created is this high cell density, structure fixed bed bioreactor with a unique design, which basically promotes a homogeneous environment within the bioreactor and a low shear environment for the cells to adhere. And this, because it's a structure fixed bed design, is scalable because all we are doing is changing the height or the diameter of the bioreactor while maintaining the same compaction in the fixed bed. This is very high capacity in a low footprint. So the highest system has 600 meters squared with the volume within a volume of 60 liters. And you can imagine what 600, and I will show actually, you need to imagine what a 600 meters square means in cell factories. And then another key benefit of this system is that it's perfect or enables continuous processing, because you can imagine that the cells are adhered or entrapped within the fixed bed, which means that you can perfuse media in and out and collect the product continuously in the case of a extracellular product. So, as I mentioned, we started with biomass immobilization for process intensification. But then we looked at the concept of chaining, which means basically having different unit operations linked together and synchronized in a way that you're minimizing the size of the surge tanks. So this started by doing it, so basically taking the bioreactor and connecting it with the TFF loop, which means that the product was being concentrated at the same time as it was being produced, and that can reduce the size of the volume being processed for subsequent steps. We've also worked to automate these steps to basically really enable continuous processing, and furthermore, we worked to integrate other steps within the midstream stage in order to ensure that whatever comes out of this platform is this concentrated, clarified bulk that is ready for downstream processing. And this translated into our product portfolio. So you have here the ScaleX bioreactors, and the ScaleX bioreactor starts with the hydro system, which is a proof of concept system, which is 2.4 meters squared. Then we move to the carbo system that is also available for clinical trial manufacture as well as R&D. This has a surface area for cell growth of 10 to 30 meters squared. And then finally, the nitro system. Now the nitro system is up to is available in two configurations. One is 200 meters squared, the other one is 600 meters squared. And this system is operated by our other leading product, which is the Nivoline system, or the Nivoline platform, which incorporates several unit operations in an automated and chain and intensified manner. And this system incorporates the bioreactor, the TFF loop, and two clarification sequences so that the product can be clarified as it's being produced while being concentrated as well. And then there is the option to have more clarification sequences prior to downstream. So as I told you, I was going to show you what this means in terms of, or equates to in terms of the cell factories. So, and also a stir tank bioreactor. So the carbo system, so the intermediate system that I've shown you, it's equivalent to tens of cell factories. So you see here 30, which is the middle between what the carbo 10 and 30 can do. And it's also equivalent to tens or even 100 actually of liters of a stir tank, depending on the process, of course. And when we look at the nitro scale, the nitro 200-600 is close to, or it equates to hundreds of multi-layer flasks and thousands, up to thousands of liters of stir tank. Of course, again, depending on the process. So now I just want to talk a little bit about how we then looked at enhancing performance through technology innovation in order to tackle that second axis, to bridge the gap in capacity in gene therapy manufacturing. So this case study was performed in partnership with 4D Molecular Therapeutics, and the objective was to look at the benefits of the structure fixed bed design. So this was a proof of concept for the production of an AAV-based gene therapy product. And here, the key highlights of this case study was that we achieved comparable productivity in VGs per square centimeter as with respect to cell factories, and and approximately fourfold higher productivity with respect to traditional packed bed reactors. Moreover, we also seen that this that during the studies, we were able to do a threefold reduction in PI and DNA usage, as well as media usage, as we reduced the density for transduction. And also we've reduced the seeding density as well, which means that we were able to reduce the seed train size. So just a couple of details on the experimental protocol that was applied. So here you see the different bioreactors that were so the different conditions in the different bioreactors that were explored. These were 4 bioreactors. The key differences was, as you see there, the inoculation, bioreactor 1 and 2, it was the same as as it was in the cell factory, but then we reduced the inoculation density by threefold on bioreactor 3 and 4. And then you also see that from the growth perspective, all of them were performed in recirculation modes. And there, what we did is that on bioreactor 4, we reduced significantly the volume of media in the recirculation loop. In terms of transfection, in bioreactor 3 and 4, we also reduced, we basically reduced by threefold the cell density at transfection, which means that we reduced by threefold the amount of PI and DNA used. And there were also changes in pH during the production stage. And finally, on bioreactor 4, we also optimized the protocol for rinsing. So we increased the number of rinses. Just on the results of this experiment, what we saw is that the cell density across the bioreactor was rather homogeneous. So when we dismantled the fixed bed and we took samples at different places of the fixed bed, you see there the difference in cell density achieved throughout the fixed beds. And then to talk about the cell growth profile, we saw a comparable cell growth profile with respect to the control, the control flasks that you see there. So you see there the kind of empty circles is basically the cell density achieved or the fold increase in number of cells achieved in the cell in the flasks versus in the scale X, which is the lines there. So you see there that the cells behave similarly in the scale X bioreactor as they did in the T flasks. And on productivity, you see the results for the 4 different bioreactors in this red kind of scale of colors versus the cell factory 10, which is the standard process, and finally the packed bed system. And you see there that in bioreactor 4, we achieved comparable results with the T flasks, even though we were reducing the seeding density as well as the transfection density. And we achieved a 4-fold higher productivity with respect to packed bed systems. When we put this into a cost model, so we made different assumptions there. We basically used publicly available information on the productivity in packed bed bioreactors and also in cell factories. And what we did is that we just use the same productivity increase ratios as we achieved in the previous case study, but with known figures. And what we got is a some results that show that this increase in productivity and performance and all the reductions in DNA, PI, and so on have a dramatic impact on the cost of goods. As for a particular dose size and annual demand scenario. So, a couple of closing remarks. Indeed, when we look at the technology landscape of traditional technologies, so considering cell factories, traditional packed bed systems, as well as stir tank bioreactors, there are several benefits of using stir tanks, so including serum-free cell culture, etc. Other benefits also include the availability in different sizes, and also the system is widely used and well characterized, and there is a lot of evidence of the use of stir tank bioreactors for different applications. Also, these systems are multi-source, so there are a number of vendors that could support customers in adopting stir tank bioreactors. That being said, there are also drawbacks to using the system. For example, if you'd like to perform a perfusion process for a gene therapy product, the use of an ATF system connected to a stir tank bioreactor might expose the cells to shear, which might not be beneficial to the cells, which makes it difficult to intensify the process using these systems. Finally, as well, it's quite difficult to remove media from the bioreactor during operations. So you would basically need to keep everything So basically, you cannot perform media exchanges or other steps that you might want to. And finally, as I mentioned, there's also large volumes being generated at the end of the process, which then need to be clarified and processed later in downstream and also midstream. But then we thought, what if we could decouple the use of suspension processes and stir tank bioreactors? So basically, decouple the process from the technology. And we did a proof of concept study using HEC-293 cells for a vaccine application, so an adeno-based vaccine application into suspension. So these were suspension cells, and we put them in our fixed bed design. And what we saw is that with a fixed bed, with the structure fixed bed design, we achieved high entrapment, equivalent and reproducible cell growth, and comparable specific cell productivity, which was very encouraging to us. So to close this session, the annual demands of gene therapy applications indeed vary significantly and can reach even 8 to the 20 VGs per year. But when we look at traditional technologies, stir tanks seem to offer the optimal technology for scalability. However, they pose large, they pose some challenges relating to the volume management after cell culture. There are newer technologies that use the principles of intensification and chaining to both increase the performance of the process, as well as reduce the operational floor space required. And an example of this is the ScaleX bioreactor and the Niveline platform that I presented today. And these dual platforms enable enough flexibility to enable customers to decouple technology from process and work on suspension and adherent cell cultures using the same technology. Thanks a lot for your attention, and please let me know if there are any questions.